It's another lecture. It's me again. Actually, it's not really a lecture. We're just gonna look at some pretty pictures, so let's just go. Here's a really cool picture I left in these notes down here so you guys could read and look at it at the same time. It's essentially undifferentiated human embryonic stem cells that are growing on a feeder layer of mouse cells. So mouse fibroblasts usually is the feeder layer. That's what we grow human embryonic stem cells and mouse embryonic stem cells on top of. So these guys, the flat elongated cells, these guys are the feeder layer, and these are the colonies of undifferentiated stem cells. So that is pretty beautiful. And we'll talk more about feeder layers versus non-feeder layers, but it's the stuff, the factors that the feeder cells are actually feeding to the embryonic stem cells that allow them to survive in culture like this. All right. So if we look at a red blood cell colony, so human embryonic stem cells that were then driven to form erythrocytes or red blood cells, right? So we have to send them down this developmental pathway, and they become this really nice, these really nice red blood cells. So again, the ability to make human blood in the lab may at some point be able to augment blood donating and all that kind of stuff, blood donation. And then this picture is pretty darn awesome. Again, derived from human embryonic stem cells. These are precursor neural cells grown in a lab. And if we look, the blue are the nuclei. So you can see the nuclei of all these guys in blue. And then the red are the actual neurons. So these long guys out here, right? And then the green are the glial cells. And remember, the glial cells are the guys that are kind of helping neurons grow up, right? And keeping them happy and, you know, I don't know, neurons. So these guys mostly out here, these are the nuclei of the glial reaching out to the neurons. And then these would be, if you see, the nuclei of the actual neurons. And the last one right here is a colony of human embryonic stem cells that are 10 months old. So they've kept them in culture for that long. Okay, sorry, that's a helicopter. I'm working outside today. Oh, jeez. I don't even know how to mute. In this case, the nuclei are green and the membranes are red. And as you can see, the nuclei are huge. So you can see the red membranes. And again, these guys, well, not again, like instead of, these guys are grown without feeder, so feeder free. So there are ways of growing stem cells without feeder layers, and that we'll get into again as we move through this course. So I just wanted to show you guys these really nice cells, little pictures, and I'll have some more for you to look at too, to get ideas of a whole bunch of different types of stem cells and what people use them for and how they actually look in the laboratory. So again, thank you for listening, and next time. Am I recording? Am I stopping? Uh-oh. Oh, there I am. So I thought I was here the whole time, but apparently I'm not. So here I am. Hi. Sorry, guys. I guess I didn't hit the video. So I'm outside. Yay. All right. Now I'm going to figure out how to stop this. Nope. Not. Whoa. That's an extreme close up. We don't want that. Here we go. Recording. Stop. Pause.